Class, this is going to be a simulation on examining genetically modified organisms. What I want you to do is the link that I gave you on Google Classroom will take you to this page. The first part of it is an explanation as to what genetically modified organisms are in comparison to breeding, like selective breeding, crosses and stuff. So please read these short explanations at the beginning, and once when you get to the end of it, please hit this virtual lab button right down here on the right side. This will take you to the virtual lab that you need to do. Now, in this virtual lab, we are going to be examining the bacterial strain B. I can't say it. You try to say it. If you say it right, bonus points. All right, so we're examining this bacterial strain. We're going to be making mutants and seeing how the genes A, B, or C, these three genes, affect the bacteria. So here you have on this page, you have the ability to turn these genes on, off, and such. Typically, mutants are organisms that don't express a certain gene. So if I was a mutant for the gene GFP, I would not express that gene where I normally would. Here, we're making a genetically modified organism. So a mutant is expressing an additional gene that it typically does not have in the wild type organism. So gene A is going to be a gene that it typically does not have. What I want you to do is to first make a mutant without any of these extra genes on. This will be your wild type standard. You always want to compare the changes in your experiment to a standard. So this is like looking at just a regular wild type bacteria. When you click the make mutant, you're going to be led to this screen, which gives you 3 different ways to observe your new organism. Culture, when you click that, it allows you to see what types of media your culture likes to grow on. This is actually very important. When you look at this, you can see these little white dots or bumps on the media. Those are colonies. This is where the bacteria can grow. You can see that examining the rich media versus the limited media, which has food X on it, the food that we're interested in looking at, the bacteria only grows on rich media. If you just see this white bump on both plates, it can grow on both. So make note of that. After culture, you can look at light microscope. Light microscope allows you to see live bacteria and see how it interacts with the environment around it. You could make notes on what they're doing from a distance and see how they interact with a conglomerate of food X. After light microscope, you could go to electron microscope. Electron microscope is a more powerful microscope. It lets you look really closely at the structure of an organism. So when you're looking at this bacteria, you can see the structure of the organism right here, very up close, a lot closer than you could on a light microscope like this. See how far away they are? And now see how close it is. Now you can make notes on the organism, what its effects are. The goal is, in this virtual lab, you're supposed to answer these questions to the right-hand side. I'm going to be making a Google document that you could answer these questions on and turn it into me. All you have to do is examine the genes that they want you to. So the first question here is, what is the function of gene A? What we examined here was a wild type organism. So what you're going to have to do is you're going to have to make a different strain with gene A and examine it under all these conditions to answer all these questions. So please go through this lab, go on Google Classroom and answer the questions in the document. If you have any questions, please ask me.